Hello everyone, my name is Nanti Leonidou and I'm a graduate student at the University of Tübingen in Germany. So today I will present you my latest project with the title Tissue-Specific Reconstruction of Constraint-based Metabolic Models based on ReconX. So almost 40 years ago, and after the first big wave of SARS, scientists warned about the potential reappearance of either the already existing coronavirus or another novel virus coming also from animals. And indeed, a year ago, this prophecy became reality with the emergence of SARS-CoV-2, leading to one of the deadliest respiratory diseases, COVID-19. However, since the beginning of this pandemic, scientists around the world try to understand the mechanism under which this novel virus spreads and adapts to new hosts, focusing mostly on detecting effective therapies and drug targets to inhibit the virus's growth. And one way to do this in systems biology is by analyzing changes in the metabolism of infected cells using genome-scale metabolic models. So in 2012, mCADRE came out, allowing the reconstruction of tissue-specific models using gene expression data and information from the network topology. However, the algorithm is implemented in MATLAB, and its functionality is based on ReconOne, the very first version of the human model. So here I present you pymCADRE, a re-implementation of mCADRE in the latest version of Python. And to test the new implementation, a gene expression dataset of human bronchial epithelial cells was used, named BEC1. And additional to that, to enable a future identification of antiviral targets against the SARS-CoV-2, host virus models were created. So table 1 compares the characteristics of the final ReconOne BEC1 specific models created by pymCADRE to the ground truth, which is mCADRE. And as it can be seen, internal optimizations done by FastFEA resulted in a model closer to the model obtained by the original implementation. However, at the cost of computational time, as FastFEA performed or needed more time to proceed. Now, since ReconOne did not include a growth function, a biomass objective function was added to the generic model prior reconstruction. And the final tissue-specific models were infected in silico with a SARS-CoV-2 viral objective function published by our group last year. And table 2 shows the analysis of the resulted host virus models using only FastCC, since FastFEA could not proceed successfully. And as you can see, FastCC operated this time significantly faster compared to the original implementation, after integrating a BOF and a VBOF. Finally, subsystem classification of the metabolic reactions in the final pruned models showed that most reactions belong to the transport subsystem, while the metabolism of cofactors and vitamins was the least represented one. Also, another positive aspect of the models created by pymCADRE is that they contain the enzyme guanylate kinase, which has been recently identified by our group as an essential component for viral growth and has a potential target for antiviral therapies. This means that the inhibition of this enzyme may be a possible target in SARS-CoV-2 therapies. Now, last but not least, the further improvements need to be done to make pymCADRE feasible with more complex and more comprehensive models like ReconII and ReconIIID. And in the future, tissue-specific models created with pymCADRE can be used to model the interaction with bacterial pathogens, having the human as host, and detect other potential antiviral targets for drugs like the guanylate kinase.